Welcome to our first day of e-learning notes. We're going to be finishing up the notes that some of you already had on Friday or Thursday before we left. Hopefully everybody is staying home and healthy and taking care of themselves, both body, mind, and spirit as well. Hopefully I'll see you all soon in person, but if not, until then, stay safe. Go Pirates. All right, today's e-learning notes are going to be titled DNA Structure and Function. The good thing about doing this via online is you can pause it, you can rewind it, and with my left hand when it gets in the way, you can take your time, fill it out correctly, not worrying about making spelling mistakes or other errors unless I'm the one that made those mistakes. So hopefully you enjoyed. Let me know what you think. If you have any questions, feel free to contact me. You can check the first e-learning folder in Schoology. There's where my office hours will be. I'll be at the computer during those 3 hours, and you should be able to get a reply pretty quickly. So go ahead and ask any follow-up questions you might have via that. If you're watching randomly online, you want to learn more about DNA structure and function, you can leave a comment. I may or may not respond to, but hopefully things get better here. All right, our notes today are called DNA structure and function. So what do we mean by structure? Well, the structure of DNA is what it looks like. And the reason why what it looks like is important is because DNA is a code. All right, and the structure of DNA determines the code that's within that DNA. Okay, so structure, what it looks like, and why is that important, and then function. Function is the job of DNA. So why do we have DNA? What is DNA? Why is it so important? Okay, so the first thing you need to know is the function. What is the function of DNA? The function of DNA is it contains coded instructions. Remember, a code is simply a way of storing information. So this is coded instructions, or some people can refer to it as a recipe, because it's not exactly step-by-step instructions. It's more of a general give or take. If you get a little bit more, a little bit less, that's okay as well. But it's the coded instructions or recipe for an organism. And how does it contain the code for that? How does the code in DNA make the organism? Well, it actually is not the code for the whole organism itself, but it is more specifically the instructions to make protein. So DNA is actually the instructions to make protein, and as we've already talked about earlier, proteins are a lot of your hormones are proteins, a lot of your transport proteins across the cell membrane. Proteins are a lot of the structures, your bones, collagen, your hair, your fingernails, those are all proteins. The colorations of your skin, the colorations of your eyes, the colorations of your hair, those are all due to the proteins inside of it. So you make those proteins make the organism. Also, proteins, as we learned about in earlier chapters, proteins are also enzymes, and if you recall, enzymes control the chemical reactions. Different enzymes control chemical reactions faster or slower, therefore it makes the organism act or behave or process things differently, and so there's where all that variation comes from, because everybody has slightly different DNA, everybody has slightly different proteins, and because everyone has slightly different proteins, everybody looks and acts a little bit differently. So really what you have is you have DNA making the proteins, and then the proteins are the little machines and the structures that make an organism. All right, so DNA is super important, because without DNA, you don't have the instructions to make the proteins, you don't have those structures, you don't have those enzymes, you don't have any of those hormones that you need. So let's talk about what DNA actually is before we get on to the function, or before we get on to the structure. What is DNA? Well, DNA is a macromolecule. Okay, remember, macro means big, molecule means molecule, a bunch of atoms combined together covalently. And so what DNA is a nucleic acid. We learned about various types of macromolecules. DNA is a nucleic acid, and that's where the N and the A come in for nucleic acid for the DNA. All right, actually DNA itself stands for deoxyribonucleic acid. If you want to sound impressive, say that out loud. Deoxyribonucleic acid. There's the D, there's the N, there's the A. We know that DNA is a nucleic acid because it says so in the name. Deoxyribonucleic acid. We'll talk in a moment about where that deoxy comes from and that deoxyribo comes from, but you do need to know that it is a nucleic acid macromolecule. It means a really big molecule. Okay? All right, so now that you know that it's deoxyribonucleic acid, we're going to talk a little bit about what it is. Well, a lot of times those macromolecules are what are called polymers. And remember, a polymer is just a long chain of repeating subunits, right? A polymer of repeating subunits. And those subunits can also be referred to as monomers. But the polymer of repeating subunits, the subunits for DNA are nucleotides. So the subunits for DNA are nucleotides. Nucleotides, nucleotides. All right, so really if you go back to when I was giving examples, if you look at a polymer as a choo-choo train, so we have a bunch of train cars following our engine. The polymer is a bunch of repeating subunits. All right, so this would be the polymer, this would be your DNA, and then these would be your monomers, your subunits, and for DNA, you would have nucleotides chained together. So that's what you guys were doing with your pop beads. You're popping them together, you're stringing those nucleotides together, making a big long chain of them. All right, now we need to talk a little bit about what the structure of a nucleotide looks like. All right, structure of a nucleotide. Okay. A nucleotide is made of 3 parts. All right, you need to know the 3 parts, but more importantly, you need to know where it aligns in this big long chain. So the first part of a nucleotide is the phosphate group. Okay, we've already talked about phosphate groups when we talked about ATP and ADP. Just like when we talked about ATP and ADP, a phosphate group is just a phosphorus and a bunch of oxygens, phosphorus and 4 oxygens. All right, attached to that, you're going to draw a line symbolizing a covalent bond and a 5-membered ring, a pentagon. Okay, so that's the first part of a nucleotide. There are 3 parts to it. Okay, and the first one is the phosphate group. The second part is the sugar. And it's a 5-membered sugar, that's why I'm drawing it as a pentagon. There are 5 carbons that make this pentagonal shape. In DNA, the sugar is deoxyribose. Okay? We know that this is sugar because it ends in ose. Most sugars end in ose: cellulose, glucose, maltose, sucrose, galactose, lactose. We've talked a bunch about those. Deoxyribose is the name of the sugar. That's where deoxyribonucleic acid gets its name, from the deoxyribose. Okay? That's not the most important part of the nucleotide. The most important part of the nucleotide is coming up, and that part is this structure right here, which is structure number 3, and that is called the nitrogen base. In the nitrogen base, there's either 1 or 2 rings. You don't need to memorize that, you don't need to know that, but you do need to know that the third part is a nitrogen base. Now, the part about the nitrogen base that's important is you have 4 possible bases. You have 4 possible bases, and those 4 possible bases are as follows. All right, you can have a base that is adenine or adenine. I've heard it pronounced both ways. I pronounce it adenine. That's the way I learned it, adenine. And we abbreviate adenine with the letter A, okay? And then we have thymine. I've also heard it pronounced thymine, but we have thymine, and thymine is abbreviated with a T, so we have A and T. And then you have cytosine, or cytosine, and we abbreviate cytosine with a C, so A-T-C. Okay? And then we have guanine. and guanine is abbreviated with a U. No, just kidding. It's abbreviated with the G. Don't put U because that'll get you confused later on because there is a base that is a U. All right, so the 4 possible bases are adenine, thymine, cytosine, guanine. All right, so we can have a nucleotide that has the same phosphate, the same deoxyribose sugar, and then adenine, we can have one that is the phosphate, deoxyribose, and thymine, the phosphate, deoxyribose, and cytosine, the phosphate, the deoxyribose, and the guanine. All right, these are named after the places that the scientists originally found them in. Adenine, I believe, was found in the adrenal gland, but don't quote me on these. Thymine in the thymus, which is a gland down here. Cytosine was found in like pus and other things. They call it a cytosol. And then guanine, I believe, was originally found in guano or bat poop. So that's where we got those names from. Adenine, thymine, cytosine, and guanine. Okay, if you need to pause, this is the first page of notes. We have one more page to go. So go ahead and pause, copy down anything you are missing, and we'll move on to page number 2. Okay, so a strand of DNA. Okay, on a strand of DNA, here's how the strands are linked together. All right, you have the phosphate group, and I'm going to just abbreviate the phosphate group as a circle, and then the sugar, a 5-membered ring, so I'll put a P there for phosphate, S for sugar, okay, and the base comes off here. Okay, so there's my first nucleotide in the strand of DNA, and then what happens is the next one attaches in the same fashion. So there's my phosphate, that sugar, and that base, and it attaches like so. So the phosphate, sugar, the sugar is the central piece, phosphate coming next, sugar coming next, and the base here is hanging off the side, phosphate next, sugar next, and that base is hanging off the side. All right, and so what you end up having is the phosphate, sugar coming down, and then those bases coming off like the teeth of a comb. Okay, so they're sticking off the side. Now, we typically refer to I'm going to switch my colors up a little bit, but we typically refer to these phosphates and the sugars because if you look at it, it's kind of like a comb. Okay, we refer to this as the DNA backbone, and so the backbone is just made of those phosphates and those sugars. All right, we usually don't draw those in because the phosphate and the sugar is always the same. It's always phosphate and it's always deoxyribose sugar. So we'll often just abbreviate this as either a line or sometimes we don't even show it at all. All right, but what is more important is these bases. We typically will show the bases as the abbreviations that we went over on the last page. So for example, this base here might be an adenine, which we'll use an A as, and this base here might be a thymine, and this base here might be another thymine, let's say, and then this base here might be a guanine. All right, so in this case, you have adenine, thymine, guanine. Now, if you notice, this part right here is the only part that changes. All right, this here is where the DNA code comes from. It's kind of like a word. That's the pattern. It's the order of the bases here. That is the DNA code. So the order of those bases is the DNA code. And then what you do is you can actually abbreviate this as well because you don't want to draw the backbone all the time. You just want to draw these bases. So you can typically just flesh in a quick backbone and then draw in those bases. So in this case, ATTG is our code, ATTG. And I'll throw even in another one here just so you guys get an idea. So you have ATTG and then C. There's my DNA code. My backbone is that phosphate and the sugars. The bases are hanging off the side. There's my code. And it's the order, and you might also hear it as a sequence, so you do need to know that order and sequence are the same thing, but right there's the code. And just like with words, they can be built of the same letters, like you can have a cat, A-C-A-T, and you can take those same letters and you can rearrange them and you can make taco out of it, which means something completely different, right? Just like in the English language, T-A-C-O, it's taco, cat, right? can mean one thing, but then you can change the order and have it be O cat cat, which means something a little bit different. So it's just like that. You have the order is where the code is. You change the order, you change the code. All right, so that's the first thing that's important. Those sequence right there is the most important part. All right, this backbone here is bonded covalently. So this is a covalent bond. We learned about that a while ago. You guys should have done a reading about that. Remember, a covalent bond is a very strong chemical bond. Okay, so that's the first part that's important. The order of the bases make the code. All right, the second thing that you need to know about DNA is that DNA is actually double-stranded. So right here on a strand of DNA, the order of the bases is equal to my code. All right, so the bases are equal to the code. Over here, DNA being double-stranded. So what I'm going to do is I'm going to bring down my ATTGC that I had over here. We have our ATTGC. This would be my coding strand. There's actually a second strand of DNA that goes alongside of it, and that second strand of DNA is called the complementary strand. It's also called the non-coding strand or the nonsense strand. Some people even call it the silent strand. It's got many different names. We're going to be referring to it as the complementary strand. And what is going to be important about this right here is this is where we're going to be learning about complementary base pairing rules. All right, so complementary base pairing rules. Okay, complementary base pairing rules are really, really simple, really straightforward. Complementary means that the bases complement each other. Not saying that they look nice, not saying that they dress nice or things like that, but they always pair up. When you have an A, for example, okay, if you have an A on strand number 1, it's always going to pair up with a T on strand number 2. Okay, so over here, this A here is going to be next to a T. Okay, then you have your T goes with your A. So anytime I have a T, T goes with an A. All right, so A goes with T, which means that then G must go with C. All right, how I remember that is 8 the, A-T, graham cracker, GC. 8 the graham cracker. A goes with T, T goes with A, G goes with C, C goes with G. Also, C and G are rounded, A and T are angular. However you helps remember it, go ahead and use that mnemonic. All right, so therefore C is going to go with G and G is going to go with C. Okay, we have a second base right here. Okay, and complementary base pairing, really easy. You have your coding strand. Whatever your coding strand is, complements it on the other strand. Now, one thing to know that is very important here with the double stranding of DNA, I'm going to draw a little dotted line here. Okay, that little dotted line, I'm doing it in red. All right, this is what is called hydrogen bonds. So hydrogen bonds hold the two strands together. Okay, hydrogen bonds are pretty strong bonds, but they're not as strong as covalent bonds. And the reason why is because this strand is going to have to open and close many times. So you want those two strands to be tightly attached so that nothing can get in there and attack your coding strand, but you want it to be able to come apart and open up. So when we start learning about replication, transcription, that DNA has to open in order to let in the code and build a new molecule on the inside to act as a template. So hydrogen bonds here, they hold those two strands together. Hydrogen bonds are bonds that hold the two strands together. But can be opened by an enzyme. And we'll talk about that later. So it opens up and then closes back up right there. So it's kind of weakly held together. strong but not too strong, kind of like a magnet or a zipper. So this is one of the reasons why the DNA kind of looks like a zipper. And also, if you look at this right here, you have your two backbones. Those are kind of like the sides of a ladder, and then the bases are kind of like the rungs. So it kind of looks like a ladder as well. And those are two versions of the models that they talk about. Okay, so the most important thing is the sequence is the bases. It's double-stranded. You have your coding strand, you have your complementary strand. The last thing and final thing you need to know for these notes is that DNA is a double helix. Okay, what that means is that that ladder that you have gets twisted around itself, and you're not going to have to memorize how many times it twists per turn. Save that for a more advanced biology class. What you get is you get your ladder right here, and it ends up twisting around itself. And this is so you can contain so much DNA in such a small little space. Every single cell in your body has the complete instructions to make you. I believe if you took all the DNA inside of yourself and put it end to end, it would go to like the moon and back a couple of times. I'm not sure exactly how many times, but each one of your cells has to contain a lot of information. And so what you do is you twist it, and as you twist it, the DNA gets shorter and shorter and shorter. So DNA is a double helix. Kind of looks like a twisted ladder or zipper, a twisted ladder or a twisted zipper. That'd be a cool band name, Twisted Zipper. And so you got your DNA as a double helix. All right, so these are the three main structures of DNA. You guys should be doing a reading about the scientists who are credited with discovering the structure of DNA. Watson and Crick, along with Wilkins and the work of Rosalind Franklin. You should be reading about that. It's a pretty interesting story. I encourage you to read further about it as well, but that goes beyond this class here. Before I sign off, there's a couple of things that I want to point out. Now, we get a little bit lazy, so typically we don't show DNA as this twisted ladder. It's really hard to draw it that way. So what we do is we show it as this singular ladder here. And then because these backbones aren't super important, what we'll often do is we'll often just show the DNA written as the bases. Okay, and so here's my coding strand, here's my non-coding strand. You can imagine the backbone is in there, but you don't have to draw it each time if you don't want to. And you can also draw the DNA sideways. Now when you draw the DNA sideways, you read it from left to right, and your coding strand is always on top. So this would be my coding strand here. It goes A, T, T, G, C. So if I draw the same strand sideways, A, T, T, G, C would be along the top, and then my complementary strand would be along the bottom. Like so. So these are the same two DNAs, just drawn different ways. You're going to see them both ways on your worksheets and your tests. Last thing that I do want to talk about goes back to the base pairing rules. Over here, we have A and T. So if your DNA is follows the complementary base pairing rules, you always know how much of every bases you have if you're just given the percentage or the amount of one base, right? Because let's say all of my DNA has to equal 100%, right? 100% of anything is all of it. So 100% is all of it. So let's say I have we'll call it complementary math. Okay. So let's say my A is 50. I can't do 55%. I'm going to do 35%. So A is 35%. We know that every time I have an A, it has to be paired up with a T. So therefore, my T is also going to be 35% because of complementary base pairing rules. So T is also going to be 35%. Right? So A and T are going to be equal. We know that C and G are going to be equal. So we can then figure out how much C and G we have, right? Because the whole thing has to add up to 100, right? So if A and T are 35 and 35, that means they take up 70% of my DNA. All right? That leaves behind 30% of my DNA to be my C and my G. So if C and G add up to 30%, that means that each one of those have to be half of that 30%, right? And 30% divided by 2 is equal to 15. So I know that 15% of my DNA is C and 15% is G. You are going to have like a test question on this, and you're also going to have a quiz question about this. But the main thing that you need to take away from here is as follows. Number one, okay, why is DNA important? Well, DNA is the encoded instructions for an organism. The DNA makes proteins, and the proteins make the organism. Okay? DNA is a nucleic acid that's made of nucleotides. Okay? The nucleotides have three parts: the phosphate group and the deoxyribose sugar. All right? And the nitrogen base. There are 4 nitrogen bases: A, T, C, and G. Those 4 bases make up the code of the DNA. The order of the sequence make up the code: A, T, T, G, C. All right? DNA is double-stranded. One strand determines the other strand. A goes with T, T goes with A, C goes with G, G goes with C. And then the last thing is that DNA is a double helix. It's twisted up on itself. All right? Pretty straightforward, pretty simple. All right? If you have questions, don't hesitate to ask me on Schoology. And I'll see you soon, Pirates.